Hello, everyone. My name is Lakshmi Nayak. I will be discussing integrating molecular targeted therapies in gliomas. These are my disclosures. Precision medicine for cancer is an innovative treatment approach tailored to the genetic profile of both patient and cancer, and targeted therapies have proven to be quite successful in oncology. The success of precision medicine is evident in many cancer subtypes, with therapies targeting BRAF, such as in melanoma, HER2/neu in breast cancer, ALK or EGFR in non-small cell lung cancer, and these therapies have transformed lives of patients with these cancers with specific molecular alterations. Additionally, small molecular inhibitors of dysregulated signaling pathways that can cross the blood-brain barrier, as well as checkpoint inhibitors and immunotherapy approaches, have also improved outcomes in patients with brain metastases from solid tumors. There has been increasing understanding of the major molecular alterations in gliomas. However, this is not translated yet in similar success in this cancer. These are some of the challenges we face for targeted therapies in glioblastomas and gliomas in particular. We lack robust representative PDX models, which may make it difficult to understand the biology or reliably predict response to drugs and in turn screen drugs. The targets as such are poorly validated. There is a lack of on-target or downstream effects, as we have noted in some surgical or phase 0 trials. Then there is the issue of the blood-brain barrier, which prevents drug from, particularly large molecules, from entering the nervous system and the tumor. While high-grade gliomas exhibit breakdown of the blood-brain barrier as evidenced by enhancing disease on imaging, all gliomas are invasive and have a component of non-enhancing disease, and this is particularly difficult to reach. Spatial heterogeneity has been described in the Transcriptional Atlas, where genomic alterations and gene expression profiles are shown to be different amongst different regions of the same tumor. And then there is the issue of temporal heterogeneity, where changes in the gene expression patterns occur from the time of diagnosis till after treatment and ultimately tumor recurrence. And this was highlighted in the phase 3 trial, Act 4 of rindapepimut. Other concerns include stem cell resistance and finally, the brain tumor microenvironment. Each tumor is unique, and within a tumor, there is diversity, which is driven by a combination of genetic, epigenetic, and microenvironmental factors. Various targets have been explored for glioblastoma, and while most have not demonstrated definitive evidence of activity in large numbers of patients, despite issues of heterogeneity and redundancy of pathways, some targets have shown activity. TKIs targeting EGFR and FGFR have not shown efficacy, and neither did targeting EGFR with peptide vaccine rindapepimut. Initial studies with mTOR and PI3 kinase inhibitors have shown either inadequate CNS penetration, like seen with sapanisertib, or if CNS penetration is achieved, as with buparlisib, there is inadequate inhibition of the pathway. There are, though, now ongoing trials of novel brain-penetrating pan-PI3 kinase inhibitor and mTOR inhibitor paxilosib, inhibitors of MDM2 such as AMG 232, inhibitors of CDK4 and 6 like abemaciclib, and inhibitors of XPO1 or exportin such as selinexor. For the purpose of this talk, I will discuss targeting BRAF, H3K27M, NTRK, and IDH, as well as the renewed interest in targeting VEGF receptor. Let's start with BRAF V600E. These mutations are known to drive cell growth in several cancers, and in brain tumors, they are most commonly found in pleomorphic xanthorastrocytoma, ganglioglioma, and some glioblastomas or grade 3 and grade 2 gliomas. Using BRAF inhibitor is a prime example of histology-independent activity of targeted therapy in a molecularly defined subset of cancer or basket trials. In this basket study of single-agent femurafenib, which is a BRAF V600E inhibitor, in BRAF V600 mutant tumors, including gliomas, there was activity evidenced in this study. Combining BRAF inhibitors with MEK inhibitors to overcome resistance mechanisms have resulted in improvement in response rate and longer duration of responses, in addition to decreased toxicities in melanoma. And so this approach, after the initial basket study of single-agent femurafenib, was also tried. And this study was presented at SNO 2019, where dabrafenib BRAF inhibitor was combined with trametinib in patients with relapsed, recurrent, or refractory BRAF mutant tumors, including high-grade gliomas. And here you can see in this study that the ORR was impressive in glioblastomas at 32%, with an overall survival rate of 82 weeks. Of course, as expected, younger patients and those with grade 3 tumors did better. Moving to H3K27M. This is a frequent histone mutation within diffuse midline gliomas and is associated with a dismal prognosis, so much so that the 2016 CNS WHO classification created a specific category of diffuse midline gliomas, H3K27M mutant. There are some novel therapies that are targeting these tumors in various approaches, such as epigenetic therapies that modify transcriptional activity at the level of the chromatin. And so this involves inhibition of the BET family proteins. Additionally, inhibitors of EZH2 activity have also been tried and shown to have efficacy in preclinical models. Agents can also be delivered by convection-enhanced delivery, including a radioactive iodine isotope conjugated to the anti-glioma monoclonal antibody 8H9. And also in these tumors, CED of pabinostat has also been investigated in a phase 1/2 study. Immune therapies, including CAR T cells against GD2, which is highly expressed in H3K27M gliomas, are currently ongoing, and so are other types of immunotherapies, such as checkpoint inhibition, as well as IDO inhibitors. And finally, molecular therapies, including small molecular DRD2 antagonists, as well as inhibitors of EGFR, have been shown in PDX models to reduce tumor growth and proliferation. I'll talk about ONC201, which is a dopamine receptor D2 antagonist. This inactivates cell signaling pathways and promotes cancer cell death through activation of integrated stress responses. There's preclinical evidence of brain penetration and activity in gliomas. There's also early evidence of clinical activity in H3K27M mutant DIPG and gliomas in adults. Moving to NTRK, which is neurotrophin receptor kinase fusions. These are oncogenic fusions involving the neurotrophin receptor tyrosine kinase genes NTRK1, 2, or 3, and coding for TRK A, B, and C, respectively. These oncogenic drivers are found in a variety of adult and pediatric tumors, supporting that basket trial approach to drug development, similar to BRAF inhibitors. In fact, NTRK fusions are now found to be clinically actionable, and the first generation TRK tyrosine kinase inhibitors, larotrectinib and entrectinib, have resulted in histology-agnostic responses in several cancers with NTRK fusion in-tract fusions. And there have also been cases of patients with brain metastases with these tumors that have responded to these drugs. Larotrectinib is an FDA-approved selective TRK inhibitor for the treatment of in-tract fusion cancers, and this has been shown to cross the blood-brain barrier. And now in a study of 14 patients with primary gliomas and brain metastases that were treated with larotrectinib, disease control without progression and partial responses were noted, and this data was presented at the 2019 ASCO meeting. Moving to VEGF receptors, while targeting angiogenesis and hypoxia and reducing tumor vascular supply is a therapeutic paradigm tried in glioblastoma for more than a decade, and bevacizumab, which is a VEGF inhibitor, has received full approval by the FDA, which demonstrated improved PFS, but did not improve overall survival. In this study, which is the REGOMA study, regorafenib, which is an oral inhibitor of multiple kinases involved in tumor angiogenesis, oncogenesis, microenvironment, and tumor immunity targeting VEGFR, PDGFR, FGFR, CSF1 receptor was tried in a randomized setting. So in this study, 119 patients were randomized to receive regorafenib or lomustine, and the overall survival was significantly improved in the regorafenib arm at 7.4 months versus 5.6 months on the lomustine arm. And these results are very impressive and encouraging. Well, in neuro-oncology, we do have a tendency to express enthusiasm when we note early evidence of activity of novel drug, and perhaps we should exert some caution while we interpret results of these phase 2 trials, importantly because we do lack validated targets. And so I thought this was a good time to bring up this editorial by Roger Stupp, where he was pointing out that the overall survival was relatively poor on the lomustine arm compared to other randomized trials. So importantly, unlike other studies which have been single-arm phase 2 trials with targeted agents, the REGOMA study was impressive because it conducted a randomized phase 2 trial, which is far more preferred than comparing to historic controls. Regorafenib now is being investigated in an adaptive randomization study as an arm for GBM Agile in recurrent glioblastoma, and we eagerly await these results and hopefully an approval. So finally, I'll talk about IDH mutations, which occur in about 70 to 80% of lower-grade gliomas and approximately 5% of glioblastomas, and are associated with increased survival. IDH mutations are early and important transformational events that remain important throughout the tumor life cycle. Inhibition of alpha-ketoglutarate-dependent dioxygenases by excess 2-HG causes genetic and epigenetic dysregulation leading to carcinogenesis. And this can be prevented by inhibiting IDH. Ivosidenib, or AG120, is a first-in-class oral reversible inhibitor of mutant IDH1 enzyme, and this has been approved for treatment of AML. A phase 1 study was conducted in IDH mutant gliomas with enhancing and non-enhancing tumors, and activity was noted particularly in the non-enhancing gliomas. Next, vorasidenib, or AG881, was tried in a phase 1 study, and importantly, this is a potent oral reversible inhibitor of IDH1 as well as IDH2 enzymes, and this is importantly also brain penetrant. In a multi-center phase 1 dose escalation study in 52 patients with gliomas that had recurred or progressed after standard of care therapy, objective response rates was 18%, and the median progression-free survival was 37 months in the non-enhancing gliomas. Based on the results of that study, there's now an ongoing phase 3 randomized blinded study of vorasidenib in non-enhancing, previously untreated IDH mutant grade 2 gliomas. There are other IDH inhibitors also under investigation which have been shown in the context of clinical trials. Finally, IDH1 mutations confer repair deficiency and PARP inhibitor sensitivity in human glioma cell lines and animal tumor models. As a result, there are now ongoing studies of using PARP inhibitors as single agents or in combination with temozolomide in IDH mutant high-grade gliomas. In conclusion, some targeted therapies clearly demonstrate activity in rare subgroups of gliomas. However, we do face challenges to development of effective therapies, primarily in the form of tumor heterogeneity and brain tumor microenvironment. Combinations are likely necessary to overcome resistance mechanisms, and efforts are currently ongoing to improve clinical trial design to accurately interpret results of early studies. And with that, I'd like to thank all of my colleagues at the Dana-Farber and from other institutions, and particularly my mentor, Patrick Wen, my funding sources, as well as patients and their families. And with that, I'll conclude and take any questions if you have. Thank you.